Hi everyone, this is Sebastian. Welcome back to the Yield Genetics podcast. And today we're diving into non-Mendelian genetics. Non-Mendelian genetics is genetics that doesn't follow Mendel's laws. To put it simply, Mendel's law of segregation says each parent passes down only one copy of each gene randomly. And the law of independent assortment says different traits are passed down separately, like shuffling cards. In classic Mendelian inheritance, dominant and recessive traits follow predictable ratios. Non-Mendelian genetics break those rules, which is what makes it so interesting. There are many different examples of non-Mendelian genetics, but I want to start with those that were demonstrated in our experiments: sex-linked genes, linkage, and epistasis. Sex-linked genes are genes located on the sex chromosomes, particularly on the X chromosome due to its larger size. Males are more susceptible to sex-linked mutations because they only have one copy of the X chromosome. Therefore, these types of recessive mutations are more likely to be expressed, and this results in a deviation from classical Mendelian inheritance patterns and in differences in inheritance patterns between males and females. In lab, we saw how white eyes were more prevalent in male Drosophila due to the white eye gene being sex-linked. Linkage is when genes located close together on the chromosome are more likely to be inherited together. Because linked genes don't necessarily segregate apart from each other and thus randomly during meiosis and are stuck together on a linear chromosome, this increases the rate of parental phenotypes. In order to unlink these genes, recombination would have to occur between them. In lab, we saw how the black and vestigial mutations were linked because the F2 cross had a greater frequency of parental phenotypes, in this case wild type or double mutants, with respect to the typical 9 to 3 to 3 to 1 Mendelian ratio for a dihybrid cross. Epistasis is a type of gene interaction when two or more genes are in the same pathway and one gene masks or modifies the effect of another. This helps us understand the order and function of genes in a biological pathway. In our lab, we crossed Drosophila with dark brown and scarlet colored eyes. The double mutant ended up having white colored eyes, which is how the two mutant phenotypes interact in an unexpected way. So far, we've talked about three key concepts: sex-linked genes, linkage, and epistasis. But those aren't the only examples of non-Mendelian genetics. There are several other mechanisms that result in deviations from the classic Mendelian ratios. Let's go through a couple more. Lethal alleles are alleles that can cause death, and when individuals with lethal alleles don't survive development, this is considered embryonic lethality. This distorts Mendelian ratios. A good example is achondroplasia, a form of dwarfism. Most people with achondroplasia are heterozygous. However, being homozygous for the achondroplasia allele is lethal. So instead of seeing the expected 3 to 1 ratio in offspring, you get a 2 to 1 ratio. And we've actually started talking about this in the context of our new module with conditional temperature sensitive mutants as a strategy to avoid this problem. Incomplete dominance is when a heterozygote has an intermediate phenotype. An easy example is flower color. If you cross a red flower and a white flower, the resulting phenotype may be an intermediate pink phenotype. This is because neither allele is completely dominant. And finally, there is duplicate gene action, which is when two different genes have redundant functions and a dominant allele of either gene results in the same phenotype. Duplicate genes are resistant to mutagenesis, and a mutant phenotype requires two independent mutations. This results in a modified 15 to 1 ratio in a dihybrid cross where either dominant allele produces the same phenotype. An example of this is seed capsule shape, which is controlled by two genes, and both must be recessive to result in a mutant capsule shape. This all ties back specifically to the flight crosses that we carried out in the lab, but there's more we could do and has been demonstrated with other models and other contexts, which is how we can learn more to understand the unexpected. Ultimately, I hope that this brief, deep dive helped illustrate the importance of inheritance patterns, how they often deviate from expected, and the relationship to phenotypes observed in humans and in nature. Thank you for listening this far. Please subscribe to our podcast if you want to hear more, and I'll see you again next time. Bye.